Hello everyone, welcome to lecture 7. So today we're going to be looking at inflammation and chemokines and the dual roles of cytokines and their systemic effects. So last lecture we looked at cytokines just for a little bit, but today we're going to have a dedicated lecture on them. So just a refresher, the response to infection is often on a continuum between innate and adaptive responses. So again, here we can see inflammation combats infection by enabling delivery of cells and mediators to infected sites by inducing local blood clotting and promoting tissue repair. And a lot of this stuff is through cytokines. So inflammation in response to infection. So cytokines can be produced by macrophages, which can cause dilation of small blood vessels. This includes certain cytokines like TNF-alpha and others. Leukocytes move to the periphery of blood vessels and as a result cause increased expression of adhesion molecules on the endothelium. Leukocytes will extravasate at the site of infection, as we can see here. And blood clotting also occurs in these microvessels. So pathogens invading tissues activate both macrophages, which release several types of cytokines and chemokines, and we'll get into more detail about these different cytokines later, which initiate local inflammatory responses. There can be dilation of local small blood vessels, which causes increased blood flow volume and decreased velocity, which can cause the heat and redness that we know about that occurs during inflammation. So you guys have probably had cuts before that might be inflamed. You can see that is red and can sometimes be warm to the touch. The endothelium can also become more permeable, where plasma proteins, including complement and fluid leakage into tissues. This is a cause of swelling. So again, bump your knee, starts to swell. This is kind of the process, what's going on. We can also have endothelial activation, where adhesion molecule expression is induced on endothelial cells, allowing for extravasation of the white blood cells. So inflammation is basically a cooperation among innate immune mechanisms. So some key mediators and events in inflammation include the vasoactive factors that are released, which influence blood vessel diameter. This includes histamine and C5a, which we discussed before. These cause capillaries to increase in diameter, causing vasodilation, and increases the permeability of the vessels. Capillaries in damaged tissues also vasodilate, which results in slower blood flow and facilitates cell exit from the blood and going from the blood to the tissue. There's also increased blood entry into the site of infection or injury, and there'll be erythema or redness, edema or swelling, and increased local temperature. Phagocytes will also migrate to the damaged tissue, following the cues from chemokines like IL-8 we've discussed before in attracting neutrophils, and C5a. Histamine and leukotriene are also released, and C5a and chemokines also contribute to the increased expression of adhesion molecules on endothelial cells, including selectins. And we'll get into these adhesion molecules later. So again, extravasation is the movement of neutrophils and monocytes out of the blood vessel, and they move into the infected tissues and is guided by chemokines and C5a. Overall, these changes in local blood vessels and endothelial cells lead to characteristic signs of inflammation, which include heat, pain, redness, and swelling at sites of infection. One thing we'll learn in another course we do in injury management, but one thing we can do to remember the signs of inflammation is SHARP. So S is swelling, H is heat, A in this scenario is loss of function, so this is kind of related to when we actually have an injury. R is redness, and P is pain. In inflammation, a series of events activates innate immune responses at a location where they are most needed. The cell recruitment is a key aspect. So here we can see a healthy mucosal surface that's not inflamed. Everything looks good. Here we have a bacteria crossing the mucosa, interacting with the macrophage. This macrophages release the cytokines. Then we'll have all our inflammatory mediators come into the tissue. We have an inflammatory reaction, and here we can see our redness, heat, swelling, and pain. So again, just a quick overview of chemokines. We discussed this earlier, but I'll just go over it again. So chemokines are chemoattractant cytokines. There are more than 50 now. They play a key role in inflammatory responses. Their main functions are to induce and control cell movement in inflammation and trafficking. They function on chemotactic gradients and also have effects on adhesion molecule expression, as we'll see later. So chemokine families are grouped by cysteine residue arrangement, as we can see here, CC family, CXC family, C, and CX3C. The CC chemokines have two adjacent cysteine residues near amino terminus, and the CXC chemokines have cysteine residues which are separated by another amino acid. These are the main two that we'll be looking at, as the majority of the more common cytokines, as you can see here, these group we'll look at quite a bit. Most chemokine receptors will bind to several different chemokines, and there's definitely redundancy in their actions. So a lot of these have overlap and they just work in different locations. So again, here's another summary figure that we've seen before. This is all the chemokines we currently know about. The main one we'll probably look at, including IL-8, which is highly involved in neutrophils. So different types of cells express different types of cytokine receptors, and this allows for specificity of chemoattraction. So certain chemokines will chemoattract certain cell types. As you can see, for example, MCP1 doesn't attract neutrophils, but IL-8 does. This is involved in monocytes. The several CXC chemokines are involved in neutrophil migration, especially IL-8, that's the main one. Several CC chemokines attract monocytes, like CCL2 or a monocyte chemoattracted protein 1. Chemokines act together with inflammatory mediators and cytokines, like TNF-alpha, to recruit cells to sites of infection and inflammation. So, some chemokines influence cell movement, as we discussed, and some attract monocytes. So the CC chemokines promote migration of monocytes and T lymphocytes, including monocyte chemoattractant protein 1 or CCL2. This attracts monocytes, including their migration from the bloodstream into the tissues, where they become macrophages. So here we can see here, a monocyte binds to adhesion molecules on the vascular endothelium near the site of infection. So here we have a chemokine receptor, so in this case it would be CCL2. And then we have our adhesion molecules, which strengthen the binding as it rolls along the endothelium. The monocyte will then migrate into the surrounding tissue, as you can see here. Then the monocyte will differentiate into an inflammatory monocyte at the site of infection and further releasing more cytokines. So circulating monocytes migrate into affected tissues. Adhesion molecules on the endothelial cells facilitate attachment to the vascular endothelium. Chemokines bound to the vascular endothelium signal the monocyte to migrate across the endothelium into the underlying tissue, and it does this by following the chemokine gradient, which is very important to reach the site of infection. This activated monocyte will produce chemokines that recruit neutrophils. So this one's initially attracted. This can then release IL-8, for example, and then attract neutrophils. So here we can see some attract neutrophils. So several CXC chemokines attract neutrophils, especially IL-8 or CXCL8, which is made by macrophages and induces neutrophils to leave the blood and migrate into infected tissues. Activated monocytes produce the chemokines that recruit neutrophils, like we just said, and chemokines have a twofold role in cell recruitment. They can act on rolling cells to promote stable binding through the effects on adhesion molecules, and they can guide migration of cells along a gradient of chemokine molecules bound to extracellular matrix and endothelial cell surfaces and infected tissues. So as we looked in the last figure, these chemokines can also work in the binding of the cells on the endothelium, but they also work in cell signaling in the chemoattracting activity. So here we can see bacteria triggers macrophages to release cytokines and chemokines. Here are cytokines and chemokines. Vasodilation occurs, and there's increased vascular permeability, which causes redness, heat, and swelling. Inflammatory cells migrate, including neutrophils, and I believe that is a macrophage, into the tissue, releasing further inflammatory mediators that can cause pain and even attracting more cells. So, how are they recruited to inflamed tissues? So, recruitment of effector cells into infected tissues occurs in inflammation. Inflammation involves the extravasation of leukocytes from the blood tissue and in a stepwise process. So, in extravasation, mediators released after tissue damage, including selectin, will have increased expression on endothelial cells. So here we can see E-selectin, which is used to bind to the cells. Here they can bind to what's called sulfated cell Lewis, or S-lex, which has a weak binding to S-selectin, or E-selectin, sorry, and it leads to reversible rolling. So when S-lex binds to the selectins, it's not completely stopped. It's slowing down, and it keeps rolling, attaching, rolling, attaching, rolling, attaching. So this is essentially just slowing it down. And then we'll see here. So before we finish this, we're just going to talk about what neutrophils can do to also bind as well. So neutrophils show shear resistant rolling. So neutrophil rolling along the vascular endothelium is a very efficient process. Neutrophils roll under high shear conditions, so high blood flow rate, where other cells can't, so they can get into places where some other blood cells or white blood cells might not be able to get to. Neutrophils have something called slings, which is a plasma membrane extension that binds to the vascular endothelium, then wraps around the rolling neutrophil. And then this tethers the neutrophil to the endothelium and can promote rapid entry during inflammation. So obviously you guys won't have this, but you can just, if you YouTube this, you can find it. So there's an electron micrograph of a neutrophil extravasating between the endothelial cells. So you can see here it's coming through. And yeah, the arrow indicates the pseudopod the neutrophil is inserting between the endothelial cells. So here we're going to take another look at rolling adhesion. So in rolling adhesion, we have selectin, which binds to S-lex interactions, which is weak and reversible and has a low affinity interaction and slows cells down. Then we have tight binding, which involves the interactions between intercellular adhesion molecules on the endothelium with integrins. So that's, you can see there's a difference between selectins, which are initially weak binding, and then our integrins, which we can see over here, which are stronger binding on leukocytes. So chemokines like IL-8, seeing that again, cause a conformational change in the integrins LFA1 on migrating cells and ligands, including ICAM1 on the endothelium. So here we can see LFA1 on the migrating cell. So this is a neutrophil binding to the other one on this endothelium, which is intracellular adhesion molecule, or ICAM. This second stage, the tight binding, will stop the cell and allows it to cross the endothelial layer. And this is through diapedesis, which you've probably heard before. So this is when cells cross endothelial wall at intracellular junctions. This is usually through proteases, which include matrix metalloproteases, which assist. Then we have migration. As you can see here, we have our first rolling adhesion, then our tight binding with our integrins, diapedesis as it moves through, and then our migration as the neutrophil follows the chemokine gradient to the site of infection. So these chemokines attract leukocytes to sites of infection and inflammation and facilitate movement into the tissues via adhesion molecules, like we said. Certain cytokines, such as TNF-alpha, induce the expression of these adhesion molecules on the vascular endothelium, including ICAM. So here we can see IL-8 causing conformational changes in the LFA, but there's something at some point is released TNF-alpha, and that causes increased expression of ICAM. So again, this allows for binding. So we essentially need both these cytokines that allow for the most efficient extravasation and binding to the endothelium. So again, inflammation is a useful localized tool in response to infection and optimizes delivery of cells and mediators to infected sites, setting the stage for the adaptive response. Inflammation at an affected site reflects the actions of cytokines and chemokines at a local level. So now we're going to take a closer look at chemokines and inflammation. So here's a kind of a good figure to just summarize the function of these three important pro-inflammatory cytokines. So these are heavily researched and you'll see them pretty much everywhere in so many studies. But yeah, we'll just take a look at the systemic effects of these pro-inflammatory cytokines. So we'll just break it down here. So IL-1 beta and IL-6, they definitely have overlapping functions, as we'll get to. So they can go to the liver and cause acute phase response, releasing acute phase proteins, including C-reactive protein or CRP, and mannose binding lectin or MBL, which we've seen before in the complement. And these are involved in activation of complement and opsonization. They can also stimulate the bone marrow to cause neutrophil mobilization. So we have stores of these white blood cells in our bone marrow, and these will release them, help with phagocytosis. They can also stimulate the hypothalamus to increase body temperature. So this increase in body temperature is important because at a higher temperature, bacterial and viral replication is less efficient, whereas adaptive immune responses operates more efficiently. This is usually because the increase in temperature results in better enzyme activity. They can also affect fat and muscle, help with protein and energy mobilization to allow increased body temperature, again for some of these beneficial effects. And we can also affect dendritic cells, where TNF-alpha stimulates the migration to lymph nodes in maturation, which is important in initiating our adaptive immune response, as we can see here. So TNF-alpha, IL-1 beta, and IL-6 have a range of actions. They coordinate responses to infection. They're involved in the activation of hepatocytes, which synthesize acute phase proteins like CRP and MBL. we have neutrophil release from the bone marrow, and these acute phase proteins act as opsonins. IL-1 beta and IL-6 and TNF-alpha also act as endogenous pyrogens, which is important, acting on the hypothalamus to raise body temperature. They also alter energy metabolism by muscle and fat. There's also a lot of exercise studies looking at these cytokines, mainly looking at how they affect fat and muscle. And there's also a lot of studies on something called adipokines, which is essentially cytokines released from fat tissue. And these are essentially related to the fact that fat cells are able to release a lot of cytokines. And especially in obesity, we can have, worse types of fat cells that release more inflammatory media, such as this, causing chronic low-grade inflammation, which is a common thing studied across several diseases. So we've seen this slide before, so I won't go into this figure, but you guys can take a look. But here we can see the acute phase response, which involves a large-scale mobilization of innate defenses. So kind of all this reaction of these three cells, also IL-1 beta, is kind of the system-wide innate defense. So the acute phase response is triggered by certain cytokines, like we discussed, IL-1, IL-6, and TNF-alpha, which are produced locally and travel systemically. Target organs of these cytokines, as we discussed, include the liver, the brain, and the bone marrow. So the acute phase proteins, as we said, are made in the liver by the hepatocytes, the liver cells, in response to cytokines released by macrophages responding to bacteria. The acute phase proteins include CRP, or C-reactive protein, and mannose-binding lectin. CRP binds to the phosphocholine on certain bacteria and fungi, but it does not bind to the form found in host cell membranes. So again, it doesn't bind to our own tissues. Both CRP and MBL act as opsonins and activate complement. CRP acts via the classical pathway by binding to C1q, essentially acting like an antibody, but in an innate way, and MBL participates and activates the lectin pathway. So again, here we can have a bacteria that induces a macrophage to produce IL-6, for example, which acts on hepatocytes to induce synthesis of acute phase protein. So again, here are some other examples, including serum amyloid protein, CRP, fibrinogen, MBL, and SPA and D. The C-reactin will bind to phosphocholine on bacterial surfaces, acting as a opsonin, activating complement, where MBL binds to mannose residues on bacterial surfaces, acting as a opsonin and also activating complement. So again, cytokines need to have a balance. So we went over this figure in our last lecture, but essentially, an overexpression of certain cytokines can have detrimental effects. For example, in COVID, we have the cytokine storm. And also in here, we have sepsis with over-release of TNF-alpha, which can cause shock. So the roles of cytokines in infectious disease. There are both pros and cons, as we just said. Cytokines are essential for defense against pathogens, yet excess cytokine of pretty much most types can also have a pathogenic effect. So many bacterial pathogens promote excessive pro-inflammatory cytokine production and can cause host damage. This includes DNF, like we just saw, IL-1, including alpha and beta, and both IL-8. Example would be gram-negatives like LPS and certain toxic producers such as toxic shock syndrome toxin, and which can cause septic shock, and toxic shock can then result, and this can be fatal. So here's just an example that sepsis is still an ongoing problem. So here we can see 1 in 3 patients with sepsis in Canada will end up dying from it. So it's about a 30% chance. This is kind of interesting because it's still extremely high mortality rate. So we can see here sepsis affects 30,000 Canadians each year, and 1/3 will die. So we don't have a completely effective response against this, and it's really dangerous once this occurs, and it's hard to reverse it. So we need to be really quick when we act. So again, here's a great paper that kind of discusses where we're at with sepsis. And again, this cytokine background will really help you understand, because it's involved in, these cytokines are involved in so many diseases that having like this solid background, this is probably one of the most important lectures in the series, because it's a good background, so when a paper is just like, oh, we saw that IL-8 did this, you can know what it does. So it allows you to just have that in-depth knowledge, even though if you're in a different field looking at obesity research, depression, because even in mental health disorders, people look at these cytokines. So they're super important. So again, here we're just going to look at innate and adaptive immune reactions. So the innate and adaptive immune aspects of the immune system do not operate in isolation. there's extensive interaction, as we've said many times. Here we can see something called the adaptive immune response, so essentially kind of the workings of them working together. So here we can see our timelines. Sorry. Yeah, with the final step being immunological memory. But yeah, we can see it working together. So we can start with our immune, as we discussed, inflammation, complement, phagocytosis, destruction pathogen. Then we have the starting of our adaptive immune response. We have our interactions between antigen and antigen presenting cells in the lymph nodes. Then we have the activation of our B cells, T cells. Then the interaction between those of them, B and T cells together, forming specific immune reactions. So kind of like we discussed with the Th1, Th2, Th17. So they will work together to figure out what's the best course of action depending on the pathogen and where it's located, such as in the mucus or in the blood. Yeah, and then here we'll try to have elimination and then memory. So how can the innate response influence the adaptive response? So one example we discussed is the activated complement proteins, which bind to bacteria, which can attach to complement receptors, or CRs as we discussed, on cells of the innate immune system. These promote opsonization by macrophages and neutrophils, and result of more phagocytosis by these macrophages ends up leading to increased antigen presentation, as you can see here, to helper T cells, and results in more T helper cell activation, which upregulates the adaptive response. We also have TLR and NOD-like receptor activation, which causes changes in gene expression in macrophage and dendritic cells. The maturation of these dendritic cells, which are the primary antigen-presenting cells, results in more effective interactions with T cells. This causes an increased expression of the co-stimulatory molecules that give positive signals to T cells. Again, we said this before, these are also called B7-1 and B7-2. But we'll talk about this later in B cell signaling and T cell signaling. We also have activation of the innate immunity through our PRRs, which leads to improved adaptive immune responses. The outcomes of TLR and NLR recognition of PAMs, bacterial apoiosis induces changes in dendritic cells, stimulating them to migrate and initiate the adaptive immunity by activating T cells. And we'll get to the function of the activation of the adaptive immune system, I believe, next lecture. So again, many cytokines produced by T helper cells control cells of the innate system. So once the adaptive system is activated, they also guide, you can see here, many of the innate cells, which are used to actually eliminate the virus. So it's not just the innate system is working, they all stop, and then the adaptive B cells and T cells come in. They actually signal back and forth to each other. As you saw in the original figure in the first lecture, these will send cytokines, T cells will send cytokines to command these cells, and these will activate these cells. So here we can see cytokines coming into play again. So cytokine releasing T cells or T helper cells can have different kinds of forms, as we mentioned, 1, 2, 17, and there's others, but they can all release certain sets of cytokines that can cause certain activation of certain cells. As we can see here, our friend IL-8, and then one unique one is IL-5 for eosinophils and etc. So we can see here that they all are very specific, so they'll know, again, if it's intracellular viruses, you'll see lots of NK cells and interferon gamma and IL-12, etc. Where if it's extracellular bacteria, you might get something else. Or if it's a parasite, you might get more eosinophils. So the interactions between innate and adaptive immunity are extensive and ongoing. It is a two-way communication. Innate responses provide protection while the adaptive response repairs. And the adaptive response can both improve and regulate innate responses. So that's the end of the lecture, guys. I hope you enjoyed the cytokines. That's probably my favorite lecture, and I think it's probably one of the more important ones. So maybe check it out again. But next week, we're going to start moving it purely into the adaptive immune system. And we'll take a look at B cell and T cell gene rearrangement, which is quite complicated, but then we'll get into their functions and stuff like that. So I hope you enjoyed it. Take care.